Welcome back to Summarize Science. Have you ever wondered how scientists can see things that are almost completely transparent, like living cells? Standard microscopes struggle with this because these samples don't absorb much light, making them nearly invisible. To see them, scientists use a special technique called quantitative phase imaging, or QPI, which can visualize these transparent structures. However, QPI systems are often expensive, complex, and not very portable. But what if you could perform this advanced imaging using technology that's already in your smartphone? That's exactly what researchers in this paper explored. They introduce a new method called quad-pixel phase gradient imaging, which uses a special type of sensor found in many modern phone cameras. This sensor is designed for phase detection autofocus, or PDAF, which helps your phone focus quickly. Here's the clever part. These sensors have a tiny microlens covering every group of 4 pixels. When light passes through a sample like a cell, it gets slightly delayed and bent. This bending causes the tiny spot of light under the microlens to shift its position. The 4 pixels underneath can detect this tiny shift by seeing an imbalance in the light they receive. By measuring these imbalances across millions of pixel groups on the entire sensor, the researchers can create a detailed phase map. This map essentially translates the invisible phase information into a visible image, all from a single snapshot. To prove their concept, the team built a custom microscope, integrating one of these commercial smartphone sensors. They had to overcome some challenges, since these sensors aren't originally designed for microscopes, but they developed clever models to correct for any issues. And the results are impressive. Here you can see their system imaging tiny, 25-micrometer plastic beads. The reconstructed phase map clearly shows the spherical shape of the beads, something a normal bright-field microscope would struggle to see with such contrast. They also discovered that using simple LED lighting, which is less perfect than a laser, actually reduces noise and makes the image clearer. It also works on biological samples. This image shows a thin slice from a mushroom. The reconstructed phase map on the right reveals the intricate, microscopic structures of the mushroom's gills with amazing detail. So, let's recap the key breakthroughs here. The researchers have successfully demonstrated a way to perform high-tech quantitative phase imaging using a common, low-cost smartphone sensor. Their method captures the entire image in a single shot, making it perfect for watching dynamic processes like living cells moving around. This technology could pave the way for a new generation of scientific tools. Imagine low-cost, portable microscopes that doctors could use for diagnostics in remote locations, or that students could use in a classroom. It could even lead to high-speed 3D imaging of biological processes, giving us an unprecedented look into the microscopic world. This work truly brings us one step closer to putting the power of a high-end laboratory into a much smaller, more accessible package. Thanks for joining us on Summarize Science.